Hello everyone and welcome to Biology with Dr. Soniyaa Shrivastava. In this class, we will be covering headings 11.5 and 11.6 part 1 of chapter Photosynthesis. We will begin the light reaction, the stage where sunlight is converted into chemical energy. We will understand how electrons move through the electron transport chain, why the pathway is called the Z scheme, and how the splitting of water provides electrons while releasing oxygen. So let's get started. Let's start with our first MCQ. Which of the following is not a direct product of light reaction? Students, before coming to the options, let's see our concept. What is light reaction? Light reaction is the power generating phase of photosynthesis. It takes place inside thylakoid membranes of chloroplast, as we have discussed in the previous video, to produce what? ATP, NADPH and oxygen as a byproduct. These products are then used in Calvin cycle, also called dark reaction, which takes place inside stroma to synthesize what? Sugars, that is glucose. Now let's see our options. Option number 1, ATP. As we have already seen that ATP is produced during the light reaction and provide energy for sugar synthesis later on. So this is not our option. NADPH. NADPH is also the product of light reaction, which provides reducing power needed in the Calvin cycle later on. So this is also not our option. O2 is produced by photolysis during light reaction. So this is also not our option. Question asks, which of the following is not the direct product of light reaction? So we have seen that glucose is produced during the dark reaction. So this is not a direct product of light reaction. So this is our answer. Moving on to MCQ number 2. In the light harvesting complex, the reaction center is formed by. Before coming to the options, let's see our diagram. Student, this is a diagram of light harvesting complex. The light harvesting complex has two parts. First, there are hundreds of pigments present here which act as an antenna to absorb light. And secondly, there is a special molecule present at the center that actually starts the photochemical reaction, and this special molecule is known as reaction center. Now coming to the options. Option number 1, a single molecule of chlorophyll B. Is it single molecule of chlorophyll B? No. Chlorophyll B is an accessory pigment. It absorbs the light and transfers the energy to chlorophyll A. Option number 3, hundreds of pigment molecules bound to protein. No. These pigments form the antenna or light harvesting complex. They absorb the energy and pass it on to the reaction center. Xanthophyll and carotenoids. This is also not our option because these are accessory pigments. They absorb the light and gives protection against excess light. So these three options are wrong. Now coming to the option number 2, a single molecule of chlorophyll A. Yes, this is our correct option. Chlorophyll A forms the reaction center and initiates the primary photochemical reaction. Moving on to MCQ number 3. In PS2, the reaction center chlorophyll A has an absorption peak at, first let's see what is PS2. As we already know that in chloroplast present the thylakoid membrane, and in thylakoid membrane present Photosystem I and Photosystem II, also known as PS1 and PS2. These two are multi-protein complex, and this multi-protein complex consists of antenna, that is SS3 pigments, which harvest the light, and second reaction center, that is chlorophyll A, which initiates electron transfer. So these two photosystems drives light dependent reactions of photosynthesis, and these two photosystems has special reaction center chlorophyll A that absorbs specific wavelength of light depending upon the specific protein bound to it. That means each photosystem has its own wavelength which depends upon the specific protein bound to reaction center chlorophyll A. Now let's see our options. First one is 700 nm. 700 nm is the absorption peak of reaction center in photosystem I. So this option is wrong. Option number 3. This lies in blue and violet region. Although pigments can absorb light here, it is not the characteristic absorption peak of photosystem II. So this is also the wrong option. Option number 4. This falls in green region, where photosynthetic pigments absorbs poorly. So it is not the reaction center peak. So this is also the wrong option. Coming to 680 nm wavelength. The reaction center chlorophyll A of the photosystem II absorbs light most efficiently at 680 nm due to specific protein bound to it. So this is our right answer. Let's see MCQ number 4, which is an application based question. What would happen if the proteins binding the light harvesting complex pigments were denatured? Denatured means destroyed. So before coming to the options, let's first see our concept. Students, suppose these are our proteins and this is the reaction center. In between these proteins are present light harvesting pigments. These are our proteins and these are pigments. Student, the pigment of light harvesting complex are not randomly scattered, but they are precisely arranged and held by the proteins. This arrangement allows the absorbed light energy to move efficiently from one pigment molecule to the other until it reaches the reaction center. So what happened if the protein framework holding these pigments were destroyed? So let's see our options. Option number 1. Light absorption would increase. Is it so? Proteins help maintain the proper arrangement of pigments. If they were denatured, light absorption and energy transfer become less, not increases. Therefore, this option is wrong. Option number 3. Water splitting would occur faster. Water splitting depends upon the functional photosystem II complex. Damaging the light harvesting complex protein would disrupt the system rather than make it work faster. So this option is also wrong. CO2 fixation would become light independent. CO2 fixation already does not directly use light, but it depends upon ATP and NADPH produced by the light reaction. So denaturing LHC proteins cannot make photosynthesis independent of light. So this option is also wrong. Now coming to option number 2. Resonance energy transfer to the reaction center would fail. Yes, this is our option. How? Proteins help maintain the proper arrangement of pigments. If they were destroyed, light absorption and the energy transfer to the reaction center would fail. So this is our correct answer. Now let's see MCQ number 5. This is assertion reason based question. What is assertion given here? The LHC contains hundreds of pigment molecules. And what is reason given here? This makes photosynthesis more efficient by harvesting different wavelengths of light. Now let's test the assertion and reason, and most important, the connection between them. So before coming to the options, let's first see what is assertion here. That LHC contains hundreds of pigment molecules. Is it true? Yes, it's true that light harvesting complex consists of hundreds of pigment molecules bound to the protein. So it's true. Now what is reason given here? This makes photosynthesis more efficient by harvesting different wavelengths of light. Is it true? Yes, it's true that different pigments absorb different wavelengths, allowing the plant to capture a broader range of light. Now ask the most important question. Why does the LHC contains hundreds of pigments? This is because different pigments absorb different wavelengths of light, allowing the plant to capture broader range of sunlight, making the photosynthesis more efficient. So reason clearly explains the assertion. Student, a plant receives sunlight containing many different wavelengths. If it had only one pigment, it would miss a large part of available light. That's why light harvesting complex contain hundreds of pigment molecules working together as a team and making the photosynthesis more efficient. So reason clearly explains the assertion. Now coming to the option. We know that our both the statement assertion is true and the reason also true, and reason clearly explains the assertion. That's why our answer is both A and R are true and R is the correct explanation. Moving on to MCQ number 6. The photosystems PS2 and PS1 are named 2 and 1 because, student, this is a conceptual trap. Let's see our options. Option number 1. PS2 works before PS1 in the Z scheme. Student, this is a correct statement that in the Z scheme, that is the non-cyclic electron transport, PS2 works before PS1, but the numbering was not based on the function. So this statement is wrong. Now see option number 3. PS2 has higher energy than PS1. While wavelength differences affect energy, but this has nothing to do with naming. So this option is also wrong. Option number 4. PS1 is only present in cyclic photophosphorylation. Number 1. PS1 participate in both cyclic and non-cyclic. And secondly, this statement is not the correct option for numbering. So this is also wrong. Now coming to option number 2. They are named in the sequence of their discovery. Yes, this is our correct option. That Photosystem II functions before Photosystem I, but the names were assigned according to the order of discovery. So this is our correct option. There is a quick memory tip. PS1 is discovered first and then PS2, but in case of function in photosynthesis, PS2 works before PS1. Let's see MCQ number 7. In the Z scheme of electron transport, the correct sequence of electron flow is, student, Z scheme of electron transport is non-cyclic photophosphorylation. So before coming to the options, let's first see what is photophosphorylation. Photophosphorylation is the process in plants that convert ADP into ATP in presence of sunlight. Photophosphorylation is of two types. First one is non-cyclic. Non-cyclic follows the linear path involving both PS2 and PS1. Secondly, this linear path starts from water and ends into NADP+. This path splits the H2O molecule, that is photolysis, to release O2 and produce ATP and NADPH. Now about the second one, that is cyclic. Cyclic follows the closed loop and it occurs only in the PS1. Here, electron leaves PS1 and return to PS1, generating ATP. In this case, only ATP is produced. Now coming to our options. The question asks, in the Z scheme of electron transport, that is non-cyclic photophosphorylation, the correct sequence of electron flow. Now student, let's see our diagram. The Z scheme diagram of electron transport, which is also known as the non-cyclic photophosphorylation, and its path is linear. It starts from H2O and ends into NADP+. Students, see here, it starts from water. Water donated electron during photolysis to the reaction center of Photosystem II to replace the electrons lost by Photosystem II. When the light hits the reaction center of Photosystem II, the electrons get excited and goes to primary electron acceptor. From there, the excited electrons move down to ETC, that is electron transport system or electron transport chain, which consists of chain of carriers, that is plastoquinone, second cytochrome, and third one is plastocyanin. As the electron pass through cytochrome, energy is used to pump the protons into thylakoid membrane and driving ATP synthesis. Now electron through ETC goes to the reaction center of Photosystem I. When light hits reaction center of PS1, again the electron re-excited and goes to electron acceptor, that is ferredoxin and NADP+ reductase. These are enzymes where electrons combines with hydrogen ion to reduce NADP+ to NADPH. Now coming to option number 1, PS1 electron acceptor and PS2. Is this option is correct? No, the electron flow never starts from PS1. So this option is wrong. It starts from PS2. Now option number 3. Electron always comes from water, not ends in water. So this option is also wrong. Now option number 4. NADP plus PS2, PS1. NADP plus is the final electron acceptor, not the starting point. Therefore, this sequence is also wrong. Now the option number 2. The correct sequence of electron flow is from Photosystem II and ends in Photosystem I, that is NADP plus, which is reduced to NADPH. So this option is correct. Now coming to MCQ number 8. Which of the following is the final electron acceptor in the non-cyclic electron transport? Student, electrons do not stay excited forever. After traveling through entire non-cyclic electron transport, they must finally be accepted by a molecule. The question is asking who gets the electron at the very end. As we have already seen in the previous question, electrons start from PS2 to goes to pheophytin, that is primary electron acceptor. From there it goes to ETC, that is electron transport chain, and these are the carriers of electron starting from plastoquinone, cytochrome B6F, and after that plastocyanin is also there. From there electron goes to PS1, and from there it goes to ferredoxin, and from ferredoxin it goes to NADP plus, which get reduced to NADPH. So question is asking which of the following is the final electron acceptor in the non-cyclic electron transport. So we have already seen pheophytin is the primary electron acceptor. So this option is wrong. Quinone, as this is electron carrier, this option is also wrong. Then option number 4, cytochrome B6F, which is also electron carrier. So this option is also wrong. Then finally NADP plus. At the end of non-cyclic electron transport, electrons are transferred to NADP plus, reducing it to NADPH. So this is the final electron acceptor. Moving on to MCQ number 9. Why is the electron transport in photosynthesis is called a Z scheme? Student, the word Z scheme does not describe where electron transport occurs. It describes how the electron flow looks when plotted on the graph. So before coming to the options, let's first see our graph. As we have already seen that how this process takes place, that is non-cyclic photophosphorylation or the Z scheme of electron transport, that it started from PS2 and goes to primary electron acceptor, then to electron carriers, then electron goes to photosystem I. After excitation it again goes to electron acceptor ferredoxin, and then finally goes to electron acceptor NADP plus and get reduced to NADPH. Student, see here that while going from PS2 to PS1 and then again to acceptor, how the electron flow takes place, what shape it takes? It takes the shape of Z. That is why we call it as a Z scheme of electron transport. Now let's see our options. Option number 1, because it follows a zigzag path in stroma. First of all, electron transport occurs in thylakoid membrane, not in the stroma. Also, the name is not based on its physical path inside the chloroplast. Therefore, this is the wrong option. Now, moving on to option number 3. Because it involves only zinc containing enzymes. There is no connection between the Z scheme and the zinc. This is a distractor. So, this option is also wrong. Option number 4. Because it ends at zone 1 of the chloroplast. Zone 1 is not a photosynthesis term, completely made up. So, therefore, this option is also wrong. Now, coming to option number 2. Based on its shape when placed on a redox potential scale. When electron flow is plotted against the redox potential, The graph forms a characteristic Z-shaped pattern. Therefore, it is called the Z scheme. So, this option is correct. Moving on to MCQ number 10. If the ETS between PS2 and PS1 is blocked, what will happen? What is ETS? ETS is Electron Transport System, or we can also say that Electron Transport Chain. So, if the ETS between PS2 and PS1 is blocked, what will happen? Student, think of the electron transport system as a bridge connecting the PS2 and PS1. Electron released from PS2 must travel through the bridge to reach the PS1. If the bridge is blocked, what happens to the electron flow? So, let's see our options. We start from option number 2. O2 evolution will stop immediately. Is this a correct answer? No. Oxygen is produced at PS2 during water splitting. This process may continue briefly, so it immediately makes this statement incorrect. So, this option is wrong. Option number 3. NADP plus will be reduced to NADPH. NADP plus can be reduced only if the electron reach the PS1. But if the ETS is blocked, the electrons will never arrive. So, this option is also wrong. Option number 4. ATP will be synthesized in excess. ATP synthesis depends upon the electron transport and proton pumping. Blocking the ETS reduces proton gradient formation. So, ATP production decreases rather than increases. So, this option is also wrong. Now, coming to option number 1. PS1 will stop receiving electrons. PS1 depends upon electrons arriving from PS2 through this ETS, that is electron transport system. If the pathway is blocked, PS1 will stop receiving the electrons and cannot function normally. So, this is our option. Moving on to MCQ number 11. This is assertion reason based question. What is assertion given here? The electrons used in PS2 are replaced by splitting of water. And what is reason given here? Continuous supply of electrons is required for photosynthesis. Student, every time when PS2 absorbs light, it loses excited electrons to electron transport chain. And if those electrons are not replaced, then PS2 would quickly run out of the electrons and stops functioning. So, where does PS2 get a fresh supply? So, before coming to the option, let's test our assertion and reason. So, what is assertion given here? The electrons used in PS2 are replaced by splitting of water. Is it true? Yes, this statement is true because water during photolysis splits into electrons, protons and oxygen. These electrons replace the electrons lost by PS2. Now coming to the reason. What is reason given here? Continuous supply of electrons is required for photosynthesis. Is it true? Yes, it's also true because photosynthesis requires flow of electrons through electron transport chain to produce ATP and NADPH. Now ask yourself, why does water split? Because PS2 keeps losing electrons and needs replacement. The continuous requirement of electrons is exactly why water acts as an electron donor. So, The reason correctly explains the assertion. Now coming to the options, both the statements are true and the reason explains the assertion. So our correct answer is both A and R are true and R is the correct explanation of A. Now moving on to MCQ number 12. In the provided Z scheme diagram, what does the vertical uphill movement of electrons represent? Now before coming to the option, let's first see our diagram. Student, in the Z scheme, electrons do not move at the same energy level throughout their journey. Twice during the process, sunlight strikes the photosystem, first Photosystem II and then Photosystem I, and gives the electron a powerful energy boost, and this boost appears as vertical uphill jumps. Now let's see our options. Option number 1, downhill flow of energy. Downhill movement occurs when electron passes through the ETC and gradually loses energy. It is not represented by uphill movement, so this option is wrong. Reduction of water. This is the wrong option because water is only oxidized, not reduced. So this option is wrong. Loss of energy is a heat. Loss of energy occurs during the downhill movement, not during the upward jump. So this is also the wrong option. Now option number 2, excitation of electrons by light energy. Yes, this is our correct option. Why? Because when photons are absorbed by PS2 and PS1, electrons gains energy and jump to a higher energy level. And this is shown as vertical upward movement in the Z scheme. That is, the excitation of electron is the vertical upward movement. Moving on to MCQ number 13. In the ETS, the movement of electrons from primary acceptor to the cytochrome complex is, before coming to the options, let's see our flow diagram. Student, once the electron is excited by light in the PS2, electron is like a ball placed at top of the hill. Does it need more energy to move or will it naturally roll down? Student, after excitation, electron reaches to primary electron acceptor and from there it moves down to different electron carriers with low energy. And this is the downhill flow. Now let's see our options. Option number 1, uphill in terms of redox potential. In the question it is asked, the movement of electrons from primary acceptor to cytochrome complex is uphill in terms of redox potential? No, because the uphill movement requires energy input. This happens only when photons excite electrons in PS2 and PS1. Between carriers, electrons do not move uphill. So this is the wrong option. Now moving on to option number 3, energy neutral. If no energy were released, ATP formation would not be possible and the electron must lose energy as it moves through the chain. So this option is also wrong. Now option number 4, powered by ATP hydrolysis. ETS helps produce ATP and it does not consume ATP to move electrons. So this option is also wrong. Now coming to option number 2, downhill in terms of redox potential. Yes, this is the correct option. After excitation, electron pass from one carrier to another with lower energy. This downhill flow releases energy which is used to pump protons and ultimately synthesize ATP. So this is our correct answer. Downhill in terms of redox potential. Moving on to MCQ number 14. Where is the water splitting complex located? Student, water splitting is not done randomly anywhere in the chloroplast. Its location is extremely important because it releases protons, and those protons are needed to build the proton gradient to synthesize ATP. Now the question is, where is the water splitting complex located? So before coming to the options, let's see the flow diagram. Student, remember the diagram of chloroplast in which stroma is the fluid filled matrix which surrounds the membranous system, and the membranous system consists of thylakoid membrane, grana. This is the outer layer of thylakoid membrane and this is the inner layer of thylakoid membrane, and water splitting complex is present inside the thylakoid membrane, and inside this lumen protons are released. So let's see our options. Option number 1, stroma. If water was split in the stroma, the released protons would remain in the stroma, but ATP synthesis requires a high concentration of protons inside the lumen. So this option is wrong. Option number 2, on the outer side of thylakoid membrane. The outer side of thylakoid membrane faces stroma. If the photolysis occurs here, protons would be released into the stroma instead of lumen. So this option is also wrong. And option number 4, in the mitochondrial matrix, this is the wrong organelle. So photosynthesis only occurs in the chloroplast. So this is wrong option. Now the option number 3, on the inner side of thylakoid membrane. Yes, this is the correct option. The water splitting complex is associated with PS2 on the lumen side of the thylakoid membrane. That is inner side of thylakoid membrane. This ensures that the released protons accumulate inside the thylakoid lumen. So this is our correct answer. Moving on to MCQ number 15. Water splitting is associated with which photosystem? Student, both PS2 and PS1 absorbs light, but only one of them has the special machinery needed to split water. This machinery is called oxygen evolving complex, that is OEC, also known as water splitting complex. So the question is, which photosystem owns this machinery? Now coming to our options. Option number 1, PS1 only. Is it the correct option? No. PS1 receives electrons and helps to reduce NADP plus to NADPH, but it does not have water splitting complex. So this option is wrong. Now option number 3, both PS1 and PS2. This is a common misconception. Both photosystem absorbs light, but only PS2 can split water, not the PS1. So this is also the wrong option. Stroma lamellae is the membrane connection between grana. They are not the photosystem, so this is also the wrong option. Then option number 2. Yes, this is our correct answer. Why? Because PS2 is directly associated with oxygen evolving complex, which splits water into electrons, protons and oxygen. So students, today we have learned how light energy drives the movement of electrons through the electron transport chain, and how the splitting of water replaces these electrons while releasing oxygen. These processes form the foundation of the light reaction and prepare the plant to produce ATP and NADPH. In the next class, we'll continue with the remaining events of light reaction, that is cyclic and non-cyclic photophosphorylation. Thank you for watching.